Okay, this is the repeat recording number 3 or 4 already. Okay, never mind, let's move on. Okay, so today we are going to talk about the associated organs or structures of the digestive system. There are 3 of them: liver, gallbladder, and pancreas. Okay, so let's go through the functions of the liver. Okay, let's start from the easiest one, then we move on to the more complicated one. Okay, so you should have learned in set 1 and 2, okay, one very basic function of the liver is it produces, okay, one of the function of the liver is it produces and secretes, okay, bile. Okay, we have already talked about what is bile in the previous lesson. So later we may touch on this again. Okay, now produces and secretes, there's a different in the meaning. Okay, produces, I think you all know, synthesize, produce something. Okay, secretes have this meaning of releases. Okay, so the first function of the liver is it will produce and secrete bile. Then the second function, okay, which is also very important, which some of you may be aware, okay, is this function of detoxification. Okay, detoxification of harmful substances. Okay, detoxification of harmful substances. Then in your syllabus, these harmful substances usually refer to alcohol. Okay, examples of alcohol, if you have learned organic chemistry in chemistry, you should know that there are many types of alcohol. Usually humans, we only drink one of them because the other alcohol are, how should I say, the side effects are too much, okay, for the human body to handle. Okay, so usually the ethanol the alcohol that we drink is ethanol. Okay, so most of the time in biology, we'll be looking at ethanol. So detoxification of harmful substances, okay, into harmless substances. Okay, so what do I mean by harmful substances to harmless substances? Okay, so the alcohol, if we go and look at it, alcohol, okay, alcohol is considered toxic. Okay, it's considered toxic to the cells, or it is toxic to the body cells. Okay, so it is considered a harmful substance. So because it's toxic, it's harmful. So the liver cells perform this very important function of converting, okay, converting this alcohol into harmless substances like water and carbon dioxide. Okay, so when alcohol are being converted, okay, converted into water and carbon dioxide, which is I would say less harmful, or in general they can be removed very easily by the body. Okay, so these are metabolic waste product. Okay, so the alcohol will be converted into water and carbon dioxide, and the water and carbon dioxide, they are considered as metabolic waste product. And these metabolic waste products, they can be removed by the body easily. Okay, they can be removed, okay, from the body easily. Okay, I think some of you are aware. So the water can be easily removed by the kidney. Okay, any excess water. Okay, any excess water will be removed by the kidney. Okay, then the carbon dioxide will be removed by the lungs. Okay, so carbon dioxide will be removed by the lungs. Okay, so this is the second function of the liver. Okay, detoxification of harmful substances into harmless substances. Okay, so I have described to you what is detoxification. Okay, to sum up, okay, so if we were to, if you need to, if you are being asked what is detoxification, bare minimum, you must tell me detoxification is a process, okay, whereby uh, toxic, harmful substances are converted into harmless substances, okay, for removal. Can I? So it will be good if you can give me examples of the harmful substances and the harmless substances. Okay, it will make your answer better. Okay, so I will put here, okay, example alcohol. Then for the harmless substances, you can put example water, okay, CO2. Okay, can I? Then, okay, the next function, the next function of the liver. Okay, let me find another place to write. Okay, the next function of the liver is to, I think this function you may have heard it in Sec 1, you may have learned this in Sec 1 and 2, Science 2. Okay, it's called deamination of excess amino acid. Now, the first concept that I want you to remember, our body do not store excess, okay? Our body does not store excess amino acid. So in the event, if we eat a lot of proteins, and these proteins are digested into amino acid, and then this amino acid are then absorbed into the blood in the small intestine, okay, are then absorbed into the blood in the small intestine. This amino acid will then be transported around the body, okay, and this amino acid will then be used by the body cells for, okay, growth and repair of cells, growth of new cells, repair of damaged cells. Okay, this amino acid may also be absorbed by the body cells to synthesize proteins, to synthesize hormones, okay, so on and so forth. Okay, so we have already learned what are the functions of proteins, or what are the roles of proteins, okay, in human. Okay, so I'm not going to write down because all these things you should have already learned. Okay, so the main point, the main thing that I want to emphasize and recap today, to emphasize today is, okay, our body does not store excess amino acid. Okay, so what will happen to this excess amino acid? Whenever I use the word excess, or whenever you use the word excess, means that after using all the amino acid that is needed, okay, what is left over? Okay, what is left over is considered as an excess. Okay, that means they are present in excess. That means the amino acid is taken in or digested after you eat the protein, and then you digest it and then you absorb it into the blood. Okay, the amount of amino acid that you have taken in, that is absorbed into the body, is more than what is needed, okay, by the body. Okay, and our body does not store this excess amino acid. So in the event where we have excess amino acid, they will be deaminated. Okay, so amino acid will be deaminated into what? Okay, it will be deaminated and then undergo a few steps. This is a few steps reaction. So I just sum up in the equation for you. So the amino acid will be deaminated into this thing called urea. Okay, and glucose will be formed in the process. Okay, so the urea will be from part of the amino acid, and the glucose will form for another part of the amino acid. Okay, so let's look at the structure of an amino acid lah. Okay, no space so I go down okay, so let's look at the structure of an amino acid. Okay, general structure. Okay, the general structure of an amino acid. Now, an amino acid is how should I say? There are different groups attached to it. Okay, so usually they will have this NH2 group. Okay, this NH2 group, okay, if I write it down for you, NH2 is called amino group. Okay, this is called the amino group. Then another group that is bonded that is found in the amino acid is called the carboxyl group. Okay, see the carboxylic acid group. Okay, to make it simple for you to understand, OH, O and H. So this is the COOH group. Okay, and this one we call it the carboxyl group. Okay, then there will be a hydrogen bonded to the carbon, then a R group. Now, this R group is the one that will differentiate the different amino acids. So different amino acid will have different R groups. Okay, so this R group will change. Okay, will be different. Okay, in different amino acid. Can I? Then maybe to make this point clearer. Okay, let me see. Let me find whether I can show you structures. Images. Structure. This will be good. Now, to make my point, I just want to actually this is not very good one. To forget that you all may not know how to read this. Copy image. Okay, I shall not use this image. I use this instead. Now, you can see here there are many different amino acids. So you have alanine, valine, leucine, okay, glycine, so on and so forth. So altogether, there are 20 common amino acids. That means there are at least 20 different amino acids that is commonly found in the nature. Okay, so if you look at a few of them, let's look at each of them. Okay, you will notice that there is this amino group. Okay, there is the carboxyl group, then the hydrogen, then this is the R group. Then if you look at another example, okay, if you look at another example, again, you have the amino acid group, the carboxyl group, okay, then this is the hydrogen, then this is the R group. Okay, this one, if I label this as R1, this is R3, then this one will be R2. Okay, the 1, 2, the subscript 1, 2, 3 basically is there to tell people the R group is different. Okay, so I use a different label for different R groups. Okay, so you can see very clearly NH2, COOH, hydrogen. Then this is the R group. Okay, maybe let's look at one more example. Okay, that is different. Okay, let's look at this one. The third one, methionine. Okay, amino acid, carboxyl group, hydrogen. This is the R group. Okay, 4, 5, 6. Okay, this is the R6, the R group. Okay, so why am I showing you this is because I need you to understand that usually the R group, okay, is made up of different atoms. Okay, different atoms, and some of these atoms are very common, some of them are not very common. Okay, so the first two common atoms that you will find in the R group will be carbon, okay, hydrogen. Then if you look here, why I show you the R6 group is because of the sulfur. So sometimes you may see sulfur atoms inside the R group. Then why suddenly got, I hope it's not your, okay, okay, it's not your. Okay, so carbon, hydrogen, sulfur, they still got one more group that is not, how should I say, it's not here. Okay, never mind. Anyway, this is not really in your syllabus. I just want to complete the story a bit. Okay, sometimes you may have phosphorus P. Okay, phosphorus, okay, that is found inside. Let me see whether I can find the phosphorus one. Hmm. Proline, it is proline structure of it. Okay, okay. Never mind. I cannot remember offhand what is the other terms are. If I'm not wrong, it's phosphorus. Okay. Anyway, it is not really in your syllabus. So for you, okay, all you need to remember, usually the R group is made up of carbon and hydrogen. Okay, the R group are a chain of carbon and hydrogen atoms usually. Okay, mainly made up of carbon and hydrogen atoms. Can I? So let's go back. So deamination of amino acid into urea and glucose. So what happened? What happened during this deamination? So let's come back to this one. So this is the structure of the how should I say of the amino acid. So this is the COOH. Then this is the H, then this is the R group. Now, so during deamination, what happened? Okay, this amino group will form. Okay, this amino group will form. Okay, the urea. Then the remaining part of the amino acid, it will form the glucose. Okay, so during deamination, okay, I know my pronunciation may not be very clear, but please don't write this animation. Okay, I always see this error. Okay, I hope it's not due to my poor pronunciation. Okay, it's deamination. Okay, can so this is wrong. This is wrong spelling. This is the correct spelling. Okay. Can so during deamination or when we say amino acid are deaminated, it basically means that the amino group, okay, will be removed from the amino acid. Okay, it will be removed from the amino acid and then it will form the urea. Then the remaining part of the amino acid will then be converted into the glucose. Okay, so this is the third function of the liver. Okay, this is the third function of the liver. Then liver still got two other functions. Let's look at the syllabus again. Okay, because this one is important. So you see, we talk about, okay, so far maybe let's copy and paste this part so that uh, I can write on it. Okay. So let's look at this one. So far we have talked about three functions, three different functions. So this is the fat digestion, is the production of bile. okay, production of bile. Then in the previous lesson, we already talked about the function of bile. Bile is important in the emulsification of fats. So large lumps of fats will be emulsified, okay, by bile into smaller fat droplets. Okay, so this is physical digestion of the fats. Then we talk about metabolism of amino acid and formation of urea. So this one is basically talking about the deamination of amino acid of excess. Okay, the word excess is important. I don't know about other bio teachers, but for me, when I mark, usually I look out for the word excess, especially if you're pure bio student. Okay, so deamination of excess amino acid. Then we also talk about the third one, detoxification. Okay, you see alcohol. So Mr. Ang know the syllabus. Okay, I hope you all can also know the syllabus. Okay, detoxification of alcohol. Okay, then we have not talked about the first one because got a lot of things to talk about. Okay, so I leave this to the last. Okay, so now let's talk about the second last function, breakdown of hormones. Okay, hormones there are many different hormones that you will need to learn in your syllabus. So in this chapter, we are going to learn about insulin and glucagon. Okay, glucagon versus glycogen, they are different. Okay, glycogen is a complex carbohydrate. Okay, this one we learn. You should learn this in the nutrients chapter. So this complex carbohydrate, glycogen. Why I want to talk about this is because. Oh, okay. Thank you. Oh. Okay. Thank you for telling me that I forget to change the screen. Okay, that's why when doing Zoom and recording cannot change here, change there. Will forget very easily. Okay. This is the part that you miss, right? Okay, the before one you managed to see, right? When I talk about the 20 common amino acid, are you able to see that? Just now you are able to see that, Okay.
Yes.
Okay, thank you. Now, so maybe I wait for a while for you to write. So far we already talked about fat digestion. We have talked about metabolism of amino acid. Okay, that means it's deamination of excess amino acid into urea. Okay, to form urea, not into urea. Then we also talk about breakdown of alcohol. So you need to learn this word, detoxification. Okay. Then now let's continue to look at breakdown of hormones. So in this chapter, you'll be introduced to hormones. So the two hormones that you're going to deal with is insulin and glucagon. Okay, and glucagon is different from glycogen. Okay, glycogen is a complex carbohydrate. Okay, this is linked to another function of the liver, which I'm going to talk. Okay, after I talk about this breakdown of hormone. Okay, and so for insulin and glucagon, okay, you all need to understand we have a lot of hormones being produced in our body. Okay, so after they have performed whatever function that they need to perform, after some time, these hormones will be broken down. Okay, and they will be broken down in the liver. Okay, full stop. That is what you need to know for this function, breakdown of hormones. Okay, so once hormones are secreted into the blood, okay, they will be transported to their target organs where they exert their effects. Then after they have done that, okay, the hormones will be broken down, okay, in the liver. Then the levels of the hormones will return back to the normal level. Okay, so since I'm talking about this, two very important concepts that I want you all to remember. Okay, hormones. Okay, usually in your syllabus, you need to remember hormones, they have a normal level. Okay, usually they have a normal level. And you need to make reference to the normal level. Okay, that means when I need this hormone to activate or to trigger or to stimulate. Okay, so basically hormones their function usually is to stimulate. Okay, stimulate target cells or organs. Okay, to stimulate target cells or organ to carry out some function, some responses. Okay, general idea that you need to remember. Hormones usually their function is to stimulate a target cell or a target organ or a target tissue. Okay, actually, I think the word tissues also. Okay, so to stimulate target structures. Okay, target structures uh, to carry out responses. Okay, then usually they have a normal level. Okay, they have a normal level. So when you need them to stimulate target cells, target organs, target structures to carry out certain responses, okay, you need to tell me very clearly, okay, whether more or less, okay, is secreted. Okay, the more and the less is important. Okay, so you must state very clearly whether there is more insulin or less insulin, or more glucagon or less glucagon being secreted. Okay, so whenever you describe, okay, the function of a hormone or how the levels of a hormone affect a process in happening in the body, you need to take note of these three things. Okay? To stimulate, okay, the function is usually to stimulate something. Okay, and insulin usually they are secreted. Uh, they are some baseline level, or we will call it a normal level. Okay, a normal level that usually will be secreted. But when the hormone is needed, is required to how should I say perform some function. Okay, to perform some function then either more or less of the hormone have to be secreted. Okay, and so you must remember these three very important functions. Okay, and now let's look at the last one. Okay, we still have 11 minutes. Let's continue. Sorry, today cannot give you a break. We are running out of time. By the way, next Thursday afternoon there will be extra classes. We need to catch up. Okay. And so let's look at the last function of the role of the liver, conversion of glucose to glycogen. Okay. Now, the last function of the liver, we call it. Why got suddenly got to tell us next Thursday? Okay, never mind. We will discuss about when to do uh, the extra lesson. Okay, can I? So let me think. What is it that I'm talking? Okay. The last function of the liver, we are looking at maintenance of maintenance of a constant blood glucose level. Okay, so again, when we talk about constant blood glucose level, okay, that is a normal level. Okay, when we say maintenance of a constant blood glucose level at what? At what level? At a normal level. At a normal level. Okay, this word is important. Okay, when we talk about the maintenance of blood glucose level, okay, you need to make reference to the normal level. Okay, so this maintenance of this constant blood glucose level is carried out by the liver. when stimulated, okay, when stimulated, okay, performed by the liver, when stimulated by, okay, more insulin or more glucagon. Okay, can I? So liver will perform this function when it is being stimulated, when the liver is being stimulated by more insulin or more glucagon. Okay, more insulin and more glucagon. Okay, just now I forget to mention something. Okay, uh, when we talk about the insulin and glucagon, they are secreted by the pancreas. They are secreted by the pancreas. Okay, and the next lesson we will talk about endocrine gland, okay, versus exocrine gland. Okay, then I will talk more about this. Let's not jump around and then get very confused. Okay, so there is a normal level of blood glucose level, a normal blood glucose level, and usually the body wants to maintain the blood glucose level at this normal level. Okay, so this is the reference point. Okay, this is the reference point. You must always make reference to the reference point. Okay, when you describe changes in the blood glucose level, you must always make reference to this normal level. Okay, can so let's look at the first scenario. First scenario, when blood glucose level increases above, when blood glucose level increases above normal level, above the normal level. Okay, what will happen? What will happen? So before we talk about it, why did the blood glucose, what will cause the blood glucose level to increase? Okay, just now I already mentioned already, I don't want to I don't want to ask people to answer question anymore. You'll take so long, waste my time. Okay, can let me see. Blood glucose level increases. So what causes this blood glucose to increase? So when we eat, okay, when we eat food rich in carbohydrate, okay, when they are digested, okay, carbohydrates like starch, when they are digested, okay, when they are digested, they will form glucose, and then this glucose will be absorbed into the blood. Okay, and this will cause the blood glucose level to increase above the normal level. Okay, this is just one example. Okay, so one possibility. Okay, so when blood glucose level increases above the normal level, okay, the pancreas will, okay, step number 2, pancreas will detect this change, and it will secrete more insulin into the blood. Okay, then this more insulin will then be transported by the blood. Okay, will be transported by the blood to the liver. Okay, then it will then stimulate the liver to absorb, okay, stimulate the liver cells to absorb, okay, more glucose from the blood, more glucose from the blood, and convert the glucose into glycogen. And convert the absorbed glucose into glycogen. Okay. This is just one function. So when when more insulin is being secreted, they will be transported by the blood to the liver. This is what is happening in the liver. Okay, actually there are other effects. Okay, more insulin got other effects, but I will talk about it in the next lesson because I want to complete the story. I don't want to leave it hanging there. Okay, so when when the liver cells absorb more glucose and then they convert the absorbed glucose into glycogen, this is what happened. So excess glucose, which is absorbed, okay, will be converted into glycogen in the liver cells. And this glycogen will be stored in the liver cells. And when this happens, okay, this will cause this will cause the glucose level to decrease back to the okay. So this will cause the blood glucose level to decrease back to the normal level. So this is what happens when the blood glucose level increases above the normal level. Okay, Ken, I think I have to stop here today already, only left 2 minutes. Okay, Ken, if any one of you have any question, okay, if you have any questions with regards to today's lesson, okay, I highly recommend that later when I share this recording, okay, go through, listen to the lesson one more time. Then can you write down the question so that next lesson you can, when we have face-to-face lesson in class, you can ask me the question. Okay, then then if you cannot wait until the next lesson or if you have my numbers, you can just PM me the question. Okay. Any question from anyone? Any urgent question? We will discuss about the extra lesson, okay, on next Thursday on the WhatsApp. Okay, I'm not going to talk about it here. Okay.